All right. Now, I'm going to put the next two categories of lipids kind of together because they have similar properties and they also have similar biological roles because these are our major membrane lipids. Now, we're going to have glycerol, and in each one of them, we're going to attach two fatty acids. But to the third carbon, we're going to have something very different than another fatty acid. We're going to have something that's quite polar. For one of these guys, we're going to have a phosphate ion. I'll represent the phosphate ion as a P with a circle on it. Now, phosphate ions have electrical charges. So of course, they're extremely polar. And then connected to that is some kind of polar molecule. There can be any of a number of different kinds of things, but their property is they are polar molecules. Okay. Now the other type of lipid, we're going to have attached to this carbon a complex polysaccharide. In other words, a chain of sugars. And I'm just making okay. Now these two molecules, of course they have different names, but many of their biological roles are quite similar. This one here is what we call, because the phosphate ion, a phospholipid. This one here, because the sugars are called glycolipids. Okay. Now, both of these are major membrane, cell membrane lipids. The majority of plant and cell and animal cell membranes, probably 90% or more, consist of one or both of these types of molecules. Now, why is that so? Well, these molecules have an interesting property here. You notice this region of this molecule here, the phosphate ion and whatever else is attached to it, that's very polar and wants to be in water. Likewise, polysaccharides are very polar. They want to be in water. So we have these polar regions. The top or head part of the molecule is very polar. Now, when we look at the rest of this, we have this glycerol and the two fatty acids. That's extremely hydrophobic. So these molecules, in effect, are kind of like AC/DC molecules. They have a polar end, what we could call the polar head, and they have a very hydrophobic tail end. So part of the molecule wants to be in contact with a polar environment like water, and the other part of the molecule doesn't want to be caught dead in a polar environment. It tries to avoid water. Now, I'm going to represent either of these kinds of molecules like so. A little circle to represent the polar head, and then these two lines representing the fatty acid tails. Okay, so polar, hydrophobic. Now, if you put a bunch of these molecules in water, the polar parts are going to want to be in contact with the water. The hydrophobic parts want to avoid water as much as possible. They can't stand being in water. So a bunch of these molecules will try to spontaneously arrange themselves so that the polar heads are facing the water and the hydrophobic tails are somehow kept out of the water. Now, one of the best ways of doing it is this. You take a double layer of these types of lipid molecules with one layer upside down or inverted relative to the other layer. Now we continue this around in 3 dimensions to make a sphere. And what we have, water in here, water out here. Now notice what we've got here, folks. The polar heads, all of them are in contact with water, both on the inside in that inner layer and on the outside in that outer layer. The hydrophobic tails, however, are shielded from the water. They're snuggled together on the inside of this structure. In a sense, it's like a molecular sandwich. You've got the polar pieces of bread, the 2 slices of polar bread on the outside, and all that nasty, greasy lunch meat on the inside. Okay, now a structure like this, once again, we take this into 3 dimensions. Water is always facing the polar heads of the molecule, which light the water. The hydrophobic tails are snuggled together away from the water. And effectively, folks, this is the foundation structure of a cell membrane. Cell membranes are a double, at least the base, the sort of foundation of a cell membrane is a double layer of lipids, what we call a lipid bilayer, with 1 layer upside down or inverted to the other. And of course, it forms a spherical shell or whatever kind of shaped shell around the particular cell, around the vesicle, or what have you. Now, artificial cell membranes, we call these things liposomes. And liposomes are used in some cosmetics. I don't know if they really do anything. But liposomes, artificial membranes, are also a potential in clinical interest because you could put some kind of drug, like a chemotherapy agent, inside there. And these can float around the bloodstream until they fuse with the cell. The stuff's not going to go into your blood. If you put something in there where they will fuse only with specific types of cells, you can deliver high-dose chemotherapy at a fairly selective manner to the types of cells you want to take down and not do so much collateral damage. So it's being investigated. I don't know if it's anything of that stuff is out clinically now, but it is being investigated as a way of delivering very high dose of chemo agents more or less specifically to the cells that you want to hurt and not the ones that you don't. In other words, doses that if you did a whole body dose of that would probably be lethal. Okay, so that's an artificial cell membrane. Of course, natural cell membranes are like this, except they have proteins embedded. More on that when we talk about cell membranes. Now, the interesting thing, though, with these lipids, these kinds of lipids, if you take these molecules, throw them into water, and then break up the clumps with like ultrasound or something, and let it sit for a while, they will spontaneously form these kinds of structures because that's the best way of keeping the polar stuff in water and the hydrophobic stuff away from water. You don't have to put any energy into it. They actually spontaneously form that way. So that's why these types of molecules are the major components of cell membranes. Now, when we look at glycolipids and phospholipids, we find that glycolipids are the major membrane lipids in plant cell membranes. Probably about 70% of the plant cell membrane membrane lipids are glycolipids. And perhaps not surprising because what do plants do? They produce huge amounts of sugars. And then they can interconvert them and make polysaccharides out. So they have a ready supply of synthesized sugars to use as glycolipids. Now, in animals, the majority of the membrane lipids in animals are phospholipids. Okay, now plants do have a significant portion of phospholipids, maybe about 20% of their membrane lipids are phospholipids. They're mostly glycolipids. Animals have some glycolipids, are relatively minor components, but they are there. And then other types of organisms may have, a lot of them have different phospho and glycolipid type things, and then some other organisms have somewhat different types of membranes. They have different compositions, but many of these molecules do have the same properties, having a polar head group and fatty and some kind of hydrophobic tail group. And they spontaneously form various kinds of membranes. Now, once again, in a real membrane to function properly, these lipids have to be able to move around. They have to be in a liquid state, not frozen solid like a chunk of piece of lard or what have you. Okay, so those are our major glycerol and fatty acid-based lipids. by doing things like making a higher portion of saturated fatty acids in these lipids. You can make a membrane that is going to be liquid only at elevated temperatures, or you replace saturated with unsaturated fatty acids in those lipids, you end up with membranes that are liquid even at fairly cold temperatures like Antarctica, stuff like that. Okay, so that's our basic things with glycolipids, phospholipids, and triglycerides. And this, I don't think we're not gonna have enough time to cover sterols today, so this is probably a good stop point, and we will see you guys in lab in about 25 minutes or so.